Okay, to keep things on time, I think we'll go ahead and start. It's my great pleasure to welcome Byron Yu, who's an associate professor of electrical and computer engineering, as well as biomedical engineering at Carnegie Mellon University. Dr. Yu studies how neurons in the brain work together to allow us to sense, to reason, and to act. In the last 15 years, advances in neural recording technologies have provided unprecedented views of the brain's activity at resolutions of individual neurons. Dr. Yu develops and applies novel machine learning algorithms that reveal how these large populations of neurons work together to give rise to brain function. His lab has two major aims: to develop and apply novel signal processing and machine learning algorithms to explain the high-dimensional structure and time course of neural population activity. The second is to apply this knowledge to the design of the next generation biomedical devices that interface with large populations of neurons. So please join me in welcoming Dr. Byron Yu.
Thank you. Oops, I should turn this on. All right, can you all hear me? Okay. Thank you for the kind invitation. This is a wonderful meeting, and I'm looking forward to interacting more with all of you. So I'm going to be talking today about brain-computer interfaces, and I put neural rehabilitation here in parentheses because this is a part of the work that I haven't done yet, but I'm very interested in. So especially for the clinicians in the room, I'd love to chat with you afterwards if you see link points between what I talk about today and your work. Okay, so this is the work of two graduate students at the time, and Patrick Sadler and Matthew Golub, who is now a postdoc, and also with Professor Aaron Batista at the University of Pittsburgh and Professor Stephen Chase, who is also at Carnegie Mellon. So what is a brain-computer interface? Okay, so it is a system whereby you record neural activity from the brain, okay, and then you pass this neural activity into some mapping, okay, which translates the neural activity into movements of a computer, of a cursor on a computer screen or a robotic arm. Okay? And on a moment-by-moment basis, the subject gets visual and perhaps other types of feedback about where the cursor is. Okay? So in this illustration, we have an animal who is moving this blue cursor using its neural activity to hit these green targets. Okay, so just to give you an illustration of how well these systems work today, I'm going to show you a video. Okay, so this is a monkey, okay, controlling this green cursor using the activity of about 100 neurons recorded in the primary motor cortex. Okay, and each time the cursor hits the target, the animal gets a reward. Right? And so movement times are typically about 1 second, okay? And the accuracy is nearly 100%. Okay? So by most metrics, the control that a subject has of a cursor, just using its brain activity, is about half as good as us moving our arms. Okay. All right. So brain-computer interfaces have received quite a bit of attention for their ability to help patients regain movement. All right. So this patient Jan was paralyzed from head to toe, okay, or from the neck down, I should say. Okay, and she was hooked up, okay, with this brain-computer interface. Right. You see this device here that's recording neural activity from the brain, okay, using electrodes implanted in the brain. Okay? And she's controlling this robotic arm, okay, to feed herself chocolate. Right? So she's just thinking about it, and the arm is moving the way she wants it to move. Okay. So this is a very important use of brain-computer interfaces. But there's another use of brain-computer interfaces, which I'd like to talk about today, which has received far less attention. Okay? And this is the use of brain-computer interfaces as both a scientific tool and as a tool for potential neural rehabilitation, okay, using neurofeedback, right? Because we can think about one of these devices as showing the subject at every moment in time what they're thinking, right? And if we can do that, maybe we can train patients who have disorders or injury to their brain, okay, which result in abnormal neural activity patterns, to train them to show normal neural activity patterns again. Okay, and this is the part that we haven't done, which I'd love to chat with clinicians about. Okay, so what I'm going to talk about today is a basic scientific study, which I believe sets the stage, okay, for these neurorehabilitation studies. Okay, so we're going to talk about learning. Okay, so whenever you learn a new skill, let's say you learn to play tennis, right, or you learn to manipulate a Rubik's Cube, right, something changes in your brain between when you're a novice and when you're an expert. Right? And so the question that we're going to ask today is why some skills are easier to learn than others. Right? And one hypothesis, right, might be that perhaps some types of neural activity patterns are easier to generate than others, right? And because some skills require certain neural activity patterns, but not others, that leads some skills to be easier to learn than others. Okay. We can go even farther and ask, can the brain learn to produce arbitrary patterns of activity across its neurons? Okay. And if not, what defines the constraints on learning to produce those arbitrary patterns? Okay, so I'll first talk about defining the key question, okay, and then I'll talk about our approach, which involves both experimental and computational components, okay, and then I'll talk about our results. Okay, so first we need a framework for quantifying neural population activity. Okay, so let us consider the situation with 3 neurons, because that's what I can draw for you. Okay, in fact, what we did in the experiment is we had around 90 units. Okay, so let each of these axes here in this space define the level of activity of one of the neurons. Okay, and so if you have the spike trains of 3 different neurons here, you can define a time window. Right? You can count the number of spikes that appear for each neuron within that window, and that defines a point in this space. Okay? We can then take many such bins, okay, and that leads to a spread of points, a cloud of points, okay, in this space. Okay. We're going to define a co-modulation pattern, okay, as a characteristic way in which these activities covary. Okay? For example, if neuron 1, every time we see neuron 1 increasing its firing rate, neuron 2 also tends to increase its firing rate. Okay? This is the sort of thing that we're trying to capture here, okay? But across the entire population of neurons. Okay, so as we already talked, learning to perform a new task likely requires a neural circuit to show new neural activity patterns. Okay, so what we're going to do here is to ask a fundamental question. Okay, what co-modulation patterns is a neural circuit capable of producing? Okay, so for example, if we observe that the neural circuit is able to produce this co-modulation pattern, is it able to reliably move its neural activity in this direction? Okay, so to be able to address this question, we need a way of directly requesting particular types of neural activity patterns. Okay, and so this is where a brain-computer interface is extremely useful. Okay, because in a brain-computer interface, what we as experimenters get to do is to define what this mapping here is. Right? So this mapping is an extremely important part of a brain-computer interface and this experiment, right? Because whichever mapping we give to the subject, right, in order for them to be able to control the cursor appropriately, right, they have to produce particular types of co-modulation patterns. Right? And then if we give them a different mapping, they're going to have to produce other co-modulation patterns. Right? So in some sense, we can now challenge the subject to show particular co-modulation patterns by designing the mapping appropriately. Okay. So previous studies, okay, have shown that different brain-computer interface systems take different amounts of time to learn. Okay? Just like if someone gives you a tennis racket, right? It's going to take you a little bit of time to get better at it. So the more practice you get with it, the better you're going to perform. Okay? And the same happens with a brain-computer interface. Okay? And what's apparent from these previous studies is that some BCI systems are more readily learned than others. Okay? And so here's the key question, right? Is there a network-level principle that explains why some BCI mappings are more easy to learn than others? Okay. So here's the approach that we're going to take. Okay. So in all the illustrations I'm going to show, okay, I'm going to show a one-dimensional brain-computer interface, okay, which means that the cursor only moves to the left and to the right. Okay? And I'm doing this for simplicity and illustrative clarity. Okay? Whereas in the actual experiments we did, the cursor could move anywhere on the two-dimensional screen. Okay? There were 8 possible targets. Okay. All right. So the first step, okay, in our experiment was to identify the region of neural activity, a region of that space in which the neural activity naturally lies. Okay? And we call this the intrinsic manifold. Okay. So how we did this was to have our monkeys passively watch cursor movements. Okay. So previous studies have reported that when you have animals passively watch cursor movements, that neural activity in primary motor cortex changes, okay, according to what the cursor is doing. Okay? So for example, if we move the cursor to the left, okay, we might see that neural activity patterns down here are produced. Okay? Whereas if the cursor is moving to the right, then these other neural activity patterns are produced. Okay? And we indeed saw the same thing in our animals. Okay. So we used this neural activity, okay, to define the space in which the neural activity naturally lives. Okay? And that we call the intrinsic manifold. Okay? And this is an empirical observation, right, that the neural activity does not uniformly populate this space. Okay? But rather, it tends to lie within more in certain areas of the space than others. Okay? And that's what we define as the intrinsic manifold. Okay? And this is presumably due to the underlying network connectivity. Right? So for example, if neuron 1 were exciting neuron 2, then we would expect their activity to go up and down together. Okay? And that would lead to particular patterns of activity being observed more frequently than others. Okay. The second step, okay, is to build a BCI mapping that the animal can use easily. Okay. So how we did this was we did the simplest thing you can imagine, right, which is we defined the following mapping. Right? So now, whenever we see a new neural activity pattern, which lies here, right, we then say, okay, the animal wants to move the cursor left, right? And then we move the cursor left. Whereas if we observe neural activity patterns here, we would move the cursor to the right. Okay? So we call this the intuitive mapping because it's intuitive for the animal to use. Okay. Now comes the key manipulation in our experiment, okay, which is to change the mapping unannounced to the animal. Okay? And the animal will have to learn to use this new mapping through trial and error. So we had two different types of changes in the mapping. Okay, the first type is consistent with the intrinsic manifold. Okay, and we call this the within manifold perturbation. Okay, and the second is inconsistent with the intrinsic manifold, and we call this an out of manifold perturbation. Okay, so to illustrate, okay, so initially we see that the animal can modulate its neural activity reliably along this axis. Okay, and then we'll switch the mapping on the animal, okay, and ask whether the animal can learn to control the cursor using this new mapping, okay, which would require the animal to reliably change its neural activity along this axis. Okay. So to illustrate the two types of perturbed mappings, okay, the first type, within manifold perturbations, as I mentioned, requires only neural activity patterns within the intrinsic manifold. Okay. So if this blue axis represents the initial mapping, okay, then the new mapping here, shown in red, remains within the intrinsic manifold. Okay? And so for the animal to control this red mapping, right, it only needs to show activity patterns within the yellow plane. Okay? And we're going to predict that this is going to be easier to learn. The second type of perturbed mapping, which we call out-of-manifold perturbations, okay, is shown here in black, okay, whereby in order for the animal to use this mapping, the animal needs to show neural activity patterns outside of the yellow plane, or outside of the intrinsic manifold. Okay? And we're going to predict that this is going to be harder to learn. Okay. So before someone raises their hand and asks, wait, isn't this obvious? Right? Why are you even doing this experiment? Right? Let me put two things on the table. Okay? The first thing is that both types of perturbations are initially difficult to control. Okay? It's not as if the within manifold perturbations is initially easy to control and the out of manifold perturbations was initially difficult to control. Right? In fact, this was one of the key controls in our experiment, which was to equalize the initial difficulty. Okay. The second thing I'd like to put on the table is that a bunch of previous studies have suggested that out-of-manifold perturbations can be learned. Okay, so I'm not going to get into the details of these studies, but several labs over the course of a couple decades even, right, because Ed Fetz, with his pioneering studies in the 70s, have suggested that these sorts of out-of-manifold perturbations can be learned. Okay, so then comes the fourth step, which is to measure the subject's ability to learn each of these types of mappings. Okay, so our central hypothesis here is that within manifold perturbations are easier to learn than out of manifold perturbations. Okay, so I'm now going to show you some results. Okay, and these experiments were done using Utah array recordings in the primary motor cortex in macaque monkeys. We typically record around 90 channels of neural activity in any given session. Okay. And we used threshold crossings. Okay, so we didn't do any spike sorting. We took spike counts in 45 millisecond bins. Okay, and we mapped the 90-dimensional neural activity to 2-dimensional cursor velocity. Okay, that's the mapping. And we use the 8-target center-out task. Okay, so let me show you a representative session for each type of perturbation. Okay, so on the vertical axis here on the left, we have success rate, okay, versus trial number within a session. Okay, so higher is better here for success rate. On the right here, we have acquire time, which is the time it takes for the animal to move the cursor from the center to the target. Okay, and here lower is better. Okay. So at the beginning of the session, the animal uses the intuitive mapping, okay, which is going to be very good at controlling, right? So success rate is very high, and acquire time is low. Okay, this is good performance. Okay, then unbeknownst to the animal, we perturb the mapping, okay, and then performance is initially bad, right? Success rate is low, right? Acquire time is high, right? And then through practice, the animal gets better at using it, right? And the success rate increases and the acquire time decreases. And then at the end of the experiment, or the session, we give the animal the intuitive mapping again, okay? And we see that the animal recovers a high level of performance. Okay. In contrast, here's an out-of-manifold perturbation. Okay? So here, what we see is that after we change the mapping on the animal, performance goes down, but it doesn't recover in the same way as it does for the within-manifold perturbation. Okay. So now if we combine the results across many sessions, okay, we can make a histogram. Oops, that went fast. Okay. So what we have here on the horizontal axis is the amount of learning. Okay, so 1 here is full learning, meaning the animal recovered the original performance with the intuitive mapping. Okay, 0 is no learning, and negative means worse. Okay, worse than before, right? And what we see here is that the within manifold perturbations showed substantially more learning than the out of manifold perturbations. Okay, and this is the key result. Okay, so what we find, okay, is that learning within the intrinsic manifold, okay, there's more learning within the intrinsic manifold than there is outside the intrinsic manifold. Okay, and this implies that what's easier or harder for us to learn depends on the existing structure of the underlying network, okay, among the neurons. Okay, and furthermore, on a time scale of hours, right, each of these sessions was about 2 hours, right, it appears difficult to generate novel neural activity patterns that are off of the intrinsic manifold. All right. So now is the more sort of forward-looking part, which we haven't done yet, which I'd like to get into, is to use this, okay, as a basis for neural rehabilitation, okay, whereby we can use this as a paradigm to try to train patients who are showing abnormal neural activity patterns to show normal neural activity patterns again, okay, by giving them, say, an appropriate mapping, okay, that requires them to show normal neural activity patterns to, say, control a cursor on a computer screen. Okay. And some sort of preliminary work that we've done along these lines, okay, has been to explore these out-of-manifold perturbations that I showed, right? All the results that I showed so far was learning within one session, okay, over the course of about 2 hours. Okay? What we're exploring now is using those out-of-manifold perturbations and allowing the animal to learn across many days. Okay? And what we're finding is that if you give the animal more time, it appears as if the animal is able to produce novel neural activity patterns away from the intrinsic manifold. Okay? And this is very promising, right? Because if we want to eventually use this for neurofeedback training, we're going to need subjects to be able to show new neural activity patterns, right, that they aren't showing naturally. Okay, that's it.